Okay, first lecture video of the new year. Um, I'm gonna play with these a little bit. Format's gonna look slightly different, I think. These may end up being shorter. Um, this unit has a lot of like processes we have to learn, kind of like math. Um, so my lectures are gonna cover like the background info and the memorization stuff. But anytime there's like a process for us to do, um, I may very quickly go over it in lecture, but then in class we will basically go over it again just in much more detail. Um, so stay tuned for all of that. I'm not in the right spot. Okay, so heredity. Um, this lecture is going to be about meiosis and general sexual life cycles. In class, we covered the second part of this, which is Gregor Mendel and Mendelian inheritance. Let's just jump right in. So, I showed this vocab page to one of my two classes already. So you may already have all this vocab down. If so, great. You can just, I'm not going to spend much time here anyway. If you haven't seen this vocab before, you do need to get all of this down. But if you have questions on what any of these are, let me know in class. I'm not going to read them out to you. Second page, again, same thing. If you have questions on what they are, let me know. But you do need to know all these vocab and have them written down. So pause if you need to. I'm going to keep going. All right, so we've already learned about mitosis, which is how cells create more of themselves. Meiosis is specific to sexual reproduction. But there are a lot of similarities to mitosis overall. The names of the phases are basically the same, but the overall outcome is quite different. Mitosis produced two genetically identical daughter cells. Meiosis produces four genetically distinct, so genetically unique, haploid daughter cells. So they have half the amount of chromosomes than they started with. So this takes place in two different stages of cell division. Meiosis 1, which is the reductional division. That's where they go from diploid to haploid, from having two sets of every chromosome to just one. And then meiosis 2 is the second division. It's the equational division. Meiosis 2 is essentially the exact same as mitosis. You just have a haploid cell that you're starting with, but it's separation of sister chromatids into two cells that are genetically unique for a reason we will talk about in just a second. So we're going to start talking about meiosis 1, which we will spend the majority of our time with. It has the most going on in it. So the first stage of meiosis 1 is prophase 1, very similar to mitosis, where its first stage was prophase. But prophase 1, basically the same stuff happens. The chromosomes condense. The only difference is they pair up in this stage. So in mitosis 1, in mitosis during prophase, we just get the chromosomes condensing. But in meiosis 1, the chromosomes condense and find their homologous partner. Remember, a homologous chromosome, they code for the same genes. One came from your father, one came from your mother, and so they pair up during prophase 1. And this is where one of my favorite topics of the year comes in. Crossing over of genes occurs in this step. I'll talk about it in more detail in a bit, but essentially, as you can see in the image on the right, they swap some DNA with the homologous chromosomes. And the result is you get two distinct, what we call recombinant chromosomes, that are a mix of your mother and father. So you don't actually pass a full mom chromosome down. You actually pass a mom chromosome with some father DNA bits in there. We'll talk more about this right now. So crossing over. This happens during prophase 1. It can kind of continue all the way up until metaphase 1. Sometimes you'll see it kind of as the in-between portion. But again, once our homologous chromosomes pair up, they physically overlap with each other and their arms like cross over each other. That's where the crossing over term comes from. And they trade portions of their chromosome, homologous portions. So they essentially like swap little gene segments. So your mom's chromosome might end up with a gene that came from your dad on it, and vice versa. We get recombinant chromosomes. So any of the chromosomes we get at the end of meiosis 1 are all completely unique. They do not exist inside your body. You don't have those chromosomes. They are genetically unique, each of them, because it's just a random amount of genes from your parents that get swapped between the chromosomes, and this leads to a huge amount of genetic diversity. We talked in class about the 73 trillion possibilities. That was not including this crossing over. Once you include this crossing over step, the number becomes incalculable. So this is a very important concept to understand. It is where a lot of genetic diversity can come from. It's where we can get weird mixes of traits that we're not expecting. And it happens in prophase 1, giving us fully unique. This is why we have 4 unique daughter cells instead of 2 copies of each unique daughter cell. They are genetic. Each daughter cell will be genetically unique. Then we get to the next step, metaphase 1, same idea as mitosis. They line up in the middle, except they are lining up with their homologous pair opposite them. So there will be one pair on the left side of the metaphase plate, one pair on the right side, or top, bottom, however you look at it. And remember, it does not matter which parent the chromosome came from. So you can have a mix of mother chromosomes on one side with fathers on one side. They don't have to all line up in order. It's just completely random. And then we attach our microtubules to the chromosomes, just like in mitosis. And from here, it's really kind of the same. Anaphase 1, we separate the chromosomes. They move towards each set. And the sister chromatids, though, are still attached. They're moving as one. So we're taking a whole chromosome instead of just a sister chromatid. Then we get to the last step of meiosis 1, which is just telophase and cytokinesis, so the cells split, and we get our 2 haploid daughter cells. A little bit of a deviation between this and mitosis is these cells are immediately going to enter meiosis 2. They're not going to duplicate their DNA or anything. And there's no need to replicate their DNA because they already have the 2 sister chromatids, so no point to do any DNA replication. We're just going to go right into the second stage. But first, just a quick overview. So we start with a diploid 2N cell, prophase 1. Chromosomes condense, pair up with their homolog. Metaphase 1, and this prophase 1 is where crossing over occurs. Metaphase 1 is where they line up in the middle, just completely random. Anaphase 1, they separate. Telophase 1 and cytokinesis, they are now haploid, and you get the 2 cells actually forming, where both cells are immediately going to go into meiosis 2. So meiosis 2 doesn't have a lot going on in it, to be honest. It's basically the same as mitosis. The only difference is the cells are haploid instead of diploid, but the actual mechanics of what happens is completely the same. Prophase 2, we get a spindle apparatus. Chromosomes should still really be condensed, and so they just start moving towards the metaphase plate. Metaphase 2, they line up in the middle, one sister chromatid on each side, but now the sister chromatids are no longer identical because of crossing over, so we're going to end up with 2 different cells at the end of this division. Anaphase 2, they separate, and then telophase, they actually arrive at the opposite poles, nuclei reform, chromosomes decondense, separate into the 2 cells. And since I started with 2, I now have 4 total. So we get 4 total daughter cells, each genetically unique and different from each other, as well as basically every other cell in your body, which is pretty cool. And that's really it for meiosis. The big stuff happens in meiosis 1. And otherwise, a lot of it is just conceptually understanding that we have reduced from diploid to haploid. We've cut the amount of genetic material in half in each cell because we need to, so that we can do fertilization later. So, meiosis 2, again, I'm not going to re-summarize this, essentially the exact same as mitosis. Nothing fancy really happens. They're haploid, though. But there are some problems that can happen here. So a big problem that can occur is something called non-disjunction, which essentially means a failure to separate. So when this happens, you get a non-disjunction event. Essentially, when you try and pull apart the two chromosomes or the two sister chromatids, you only end up pulling one. And so the result is you'll end up with cells where some of them have too many chromosomes and some of them have too few. When you don't have the right number, we call this aneuploidy. It just means you have an abnormal number of chromosomes. And in plants, this is actually pretty common. Plants kind of handle it just fine for whatever reason, but animals, it is usually a fairly big event. It can be fatal or lead to developmental disorders such as Down syndrome, which is trisomy 21, meaning you have 3 copies of chromosome 21 instead of 2, which leads to the case of Down syndrome. So aneuploidy can cause problems, and it results from this non-disjunction failure to separate events where when we're doing anaphase, we don't actually separate the two parts that we're supposed to, and they stay stuck together. You can see that in the image on the right. So some quick comparisons with mitosis, then we will wrap up. It is good to know the differences. So when does DNA replication occur? In mitosis, it's during interphase. In meiosis, it's also during interphase. So same there. Number of divisions, 1 for mitosis, 2 for meiosis. So there's double the amount of divisions in meiosis. Synapsis of homologous chromosomes, that's when your homologous chromosomes pair up and do the crossing over thing, only happens in meiosis. How many daughter cells, and are they genetically unique? 2 genetically identical daughter cells for mitosis. 4 genetically unique haploid daughter cells. And then lastly, the role of it, as we know, growth and repair is mitosis's main feature, whereas meiosis is purely for the production of gametes. Reducing your chromosome number allows you to do a fertilization event, a union of sperm and egg, that again helps you increase your genetic variability to make you more fit. So the 3 events that are unique to meiosis that you should know is that in prophase, there's crossing over. In metaphase 1, homologous chromosomes line up instead of individual chromosomes, so they're pairs of chromosomes instead of individuals. And then in anaphase 1, homologous chromosomes instead of sister chromatids are the things that separate. So those are the 3 key differences. Everything else is basically the same. And that is it. We are now to where we were in class. Again, as we get more into it, we will be doing a bit more lecture style things in class. But for now, please let me know if you had any questions on this, and I will see you in class. That's all. Farewell. See you later. Bye.